Hi, everybody. Welcome back. So we can basically turn off or lose the tumor suppression power of a tumor suppressant gene by breaking that gene. And we can break that gene either by having a mutation in the gene that renders it inactive. We might have chromosome loss, which would contribute if the piece that's missing actually carried a piece of our tumor suppressor gene, right? And or you can methylate your tumor suppressor gene. And abnormal methylation of CPG islands near promoter regions tend to do this, and they tend to have that effect. They tend to kill your tumor suppressor genes when that happens. So you want to protect your CPG islands as much as you can. So shifting gears, just a little, well, not shifting gears, but we've been talking about how to prevent cancer, right? We have these tumor suppressor genes we've been talking about. We've been talking about different types of tumors you can get. And so, let's talk about cancer. Cancer is uncontrolled cell division, and it happens in a series of steps. Typically, this has to happen to the same cell in order for it to actually result in cancer and metastatic cancer. So you'll have a benign alteration to your genome in the form of a mutation. And our bodies have evolved in a regime where mutations are happening at a constant rate as a function of cell division, as a function of time. And so we have ways of dealing with that. So that's not a huge problem. It always begins with a benign genetic alteration, but we have ways of dealing with that. Then over time, these mutations accumulate and we break genes. We break the function of genes, and then we start to break the function of oncogenes, right? So we start to break the function of very important genes that are involved in the regulation of the cell and preventing the cell from progressing in the cell cycle, ultimately into cell division, right? Because what we don't want with a cell that's got messed up genes is copies of that cell running around in the body. That's exactly what cancer is. Okay. Over time, with additional mutations, that cell that has faulty oncogenes, broken oncogenes, broken tumor suppressor genes, that's when it becomes malignant. And your cell has now accumulated enough changes that the checking software of the cell is broken, is disturbed. And then once your cell is malignant, it can continue to accumulate genetic changes, which means you start to have breakdown in cell function in all new fun ways. So the disease is complex. It's very complex. There's many symptoms that can manifest because it started off with just being a cell that divided when it wasn't supposed to, and now it's perhaps degrading proteins it's not supposed to. Perhaps it's taking more energy and burning up more glucose than other cells around it. It's producing more ATP, or it's undergoing fermentation and not glycolysis more than other cells, right? And these are all going to impact your physiology, your metabolic processes, and your whole body. So it's a difficult disease to sometimes identify because the symptoms can be mysterious. Lung cancer is diagnosed in approximately 170,000 people each year. It's a large problem. Worldwide, more than 1 million people are diagnosed every year. And the reason we're talking about it is because 90% of the cases are caused by smoking and are thus preventable. So we have to talk about it, because it doesn't have to happen. It doesn't have to be in your future or your loved ones' futures. Unlike other cancers for which early diagnosis is possible, lung cancer is usually detected late, and it's detected after it's metastasized, after it's quite advanced. And that makes it really difficult, if not impossible, to cure. And a 5-year survival rate, once you've been diagnosed with lung cancer, drops down to 15%. It's a really tough diagnosis with not great disease outcomes. Okay, so let's talk about what happens to you. Most of your lung cancers are considered carcinomas, and that is medical jargon for epithelial cells that have cancer. Your mutations accumulate at first in what are called basal cells, and their numbers increase, and having a bunch of basal cells is not normal, so we call it hyperplasia. And then as these mutated basal cells accumulate, they develop abnormal morphologies. And that abnormal morphology has its own name called dysplasia. This is all important because it's relevant. We'll come back to these terms when we talk about histology and tissues. Okay, so in the early stages, the abnormal basal cells are considered precancerous. And so that's the moment when there's a possibility that you can right the ship, right? They haven't metastasized. It's not chronic yet. They're there, but your body may be able to fight them off and create apoptosis in those living cancerous cells before it gets too late. Or you can keep smoking. You can keep being exposed to nicotine and cigarette smoke. And if that's the case, then these abnormal cells accumulate and additional genetic changes accumulate. And then we get uncontrolled cell division. Once this happens, the patient has basal cell carcinoma, and metastasis is when these cells travel to other parts of the body, and they basically spill into the bloodstream and are able to circulate throughout our tissues, and they land in some brand new place, and they continue to divide in an uncontrolled fashion and cause issues wherever they land. Okay, so here's an image of the basal cells. So this is your typical lung cross section. You've got columnar epithelial cells, and then you've got basal cells, basal being bottom. They're cells that are found underneath these columnar epithelial cubular shaped cells. And they have a slightly different function than our columnar epithelial cells. We'll talk about this again when we get to tissue types, but these basal cells are right against the basement membrane, and then the basement membrane is what is going to be attached to other cells which make up our connective tissues. Okay, if we were to look at an actual stained magnification, this is what it would look like. So the basal cells are here in the dark color, and the connective tissues down here. Basement membrane is this kind of striated pattern that's here, and then our columnar epithelial cells are here. Hyperplasia is the proliferation of those cells, and here you can see them. They've been, they're yellow, so there's a whole bunch. They have a different shape than your columnar epitheliums. They look like little stacks of books, and they're column-like. Your basal cells are not. They're more globular, and they're not supposed to be a lot of them. But this is an indication of a person with something wrong going on, right? If you had this many basal cells in your lung tissues. And you can see that the columnar epithelium is really pushed up, and that's hyperplasia. Then you get the loss of ciliated cells. So our columnar ciliated cells are just completely compacted and smooshed down because of all of these basal cells that have built up between the epithelium and the basement membrane. And that's what it would look like in real life here. I mean, look at the texture, right? Look at the difference. And these, right, our columnar epithelium cells are there to help us have gas exchange. And so they have to be tall and long like books. This is not going to have very good gas exchange. Okay, you're not going to breathing's going to be really messed up in this person. This is initially the dysplasia precancerous stage, and then it progresses to cancerous. And what we see is metastasis. So we see our basal cells just kind of pushing and spilling out of the place where they were into other parts of the tissue. So they're in the connective tissue now. They pushed past the basement membrane. They've completely morphed and bubbled this whole region here. So that's your typical progression. I hope that was sufficiently upsetting and scary enough to motivate you to quit smoking if you are a smoker, and to tell your loved ones, if they're smokers, that they should consider quitting, because it really is, lung cancer is preventable, and the probability that you get lung cancer as a smoker is very high. Okay, that's it for now.